Hello everyone. So for my project, to study the tissue heterogeneity and tissue-specific function of AMP, I evaluated the existing isoforms that were found to occur naturally in the rodents. So these are termed as the isoform 1, 2, and 3 in my project as shown in the top left corner of this slide. They exist due to alternative splicing of the full-length wild-type MPRA receptors, and the mutation are shown by the crosses. So as such, to evaluate them, I created these isoforms using plasmid and site-directed mutagenesis, which I then transfected and expressed the receptors on the surface of a mammalian cell lines. Since MPRA has an intracellular guanylyl cyclase domain, upon binding of AMP, cGMP is being produced. Hence, adding AMP to cells that are expressed with the receptors, the cGMP production can be used as the readout. So I evaluated cells that are transfected with the splice isoform alone, or when it is being co-transfected with the full-length wild type using various ratio that actually reflects the ratio that is detected in the rodents. So GFP efficiency tests, Western blotting, as well as immunofluorescence were techniques that were used to actually confirm that these receptors are being expressed on the cell surface of the mammalian cell line. My result actually shows that clearly that the isoforms actually reduces the cGMP production of the wild-type MPRA even at a low percentage of the total MPRA receptors. So this actually suggests that the isoforms do play a role in its tissue specificity of AMP. Another part of my project which looked into the tissue heterogeneity of AMP in a renal nephron. This is shown on the right side of this slide, and AMP is known to actually induce vasodilation in the afferent arterioles AA and vasoconstricts the efferent arterioles EA. And interestingly, these distinct responses are governed by the MPRA receptors alone. However, the responses towards the two AMP analogs created by our lab were not evaluated. These two AMP analogs were designed such that the DGD AMP analog actually retains only the renal function, while DRD AMP retains only the vasodilatory function with no renal function. For this project, I went on a research exchange to Germany to master the technique of isolating the renal afferent and efferent arterioles from mice kidney and perfuse the vessels. This isolation techniques is actually quite hard in terms of it require a very steady hand, lots of patience, and long duration under the microscope, as the blood vessels are actually tiny such that it is about 3 to 5 micrometer in length and about 1 micrometer in thickness. So the perfused vessels were subjected to either AMP or the two AMP analogs, and the changes in the thickness were then determined using a video stereo microscope. Our result actually shows that the DGD AMP behaves similarly like AMP, where it actually induces vasodilation in the afferent arterioles and vasoconstricts the efferent arterioles. Whereas for DRD AMP, it does not alter the thickness of the efferent arterioles, but it weakly constricts the efferent arterioles. So this is aligned to their function that was found in vivo and in vitro study, and which actually promise to study further into the mechanism that is involved in this upholding the tissue heterogeneity in the kidney. We might also be able to find out why a vasodilator like AMP do also causes vasoconstriction in the efferent arterioles. So for this, I would like to thank you for listening to my video.